Okay, we'll get started. PA students, tomorrow you have 8 a.m., you have history taking at 10 o'clock. Dr. Wood's going to come in and do pharmacodynamics at 10, and you guys will be done in the afternoon. Okay? I will give you the option to, we only have like 90 slides to get through GI, so we won't need the full 4 hours today. So I will give you guys the option to think about when we finish. Do we want to do the review today? It's just a Jeopardy review, knock it out. Then Thursday you don't have class? Yes. Okay. Okay, so let's get through the GI stuff. See what we can get through. The GI system is very straightforward. These are the objectives for today. So, the GI system, when you are studying the GI system, the way you do it easily is to just pretend you're eating something and follow it through the entire way down the system until it leaves your body. So if you're taking a piece of food and you start chewing on it, that starts the digestive process. So think about the food that's in my mouth, what's going on in the mouth. Then I swallow, it's now in the esophagus. So what is happening in the esophagus? What's important about the esophagus? Then move into the stomach. So now the food's in the stomach. What is going on in the stomach? What is the stomach doing? Then, okay, fine. Now the food bolus is leaving the stomach, it's going to the small intestine. What's happening in the small intestine when the food gets there? That's how the easy way to study this material is. Just pretend you're a piece of food, follow it through the entire system on the way out. What's happening in each different anatomical part. The easy way to think of the GI system is just one long tube. From the mouth to the anus, just one long tube, and it has some accessory organs. The salivary glands in the mouth, liver, pancreas, gallbladder, dump in more things, but it's all one long tube. There are some major functions that the GI system is responsible for. The reason why we talk about it this way is because when it comes to GI disorders, it's going to fall under these categories. So the first major function for GI system, motility. Movement of food through the GI system. If the movement goes too slow, what do you get at the end there? Constipation, it's going too slow. If it goes too fast, diarrhea. So you see how you have motility issues. Diabetics, they have what's called gastroparesis, where the stomach doesn't really work very well, the musculature of the stomach doesn't work well, and the food sits there for a long time. That's slow motility, nothing's really going on. So motility, is it going too fast or too slow through the system? If the food goes too fast, you don't have enough time for absorption to occur. We call it absorption here because this is the first time we are absorbing these nutrients. In the kidneys, it's reabsorption because we've already absorbed them from the environment. And in the kidneys, we're just going to reabsorb them back into the system when they got pushed into the filtrate. Next one, secretion, appropriate fluid and enzyme solutions there should be more there due to the macromolecules. We eat carbohydrates, fats, and proteins. They are macromolecules, they are big molecules. We have to break them down into the smaller parts. Secretion helps that process occur. Digestion, this is the actual part of breaking them up. So digestion, from a physiological standpoint, is breaking down macromolecules into the smaller ones. All those complex carbohydrates into glucose, proteins into amino acids, and all the cholesterol, like the fat, is broken down into fatty acids and glycerol that we can then take up. So that is the digestion, breaking it down into smaller parts. Then we have absorption. Can we absorb them into the circulatory system? Okay, some people have We'll talk about it in a little bit, like the celiac disease or these gluten sensitivities. There's more to it than just the gluten, but what happens is you pretty much get rid of all the microvilli, the lining in the small intestine with these types of absorption problems. So it takes about a week for all that villi to regrow again. So if you have like lactose intolerance and you drink some milk and then you have really bad stomach problems, it's going to take about a week before you're fully recovered because you have to regrow the lining of the intestine that you just destroyed by drinking whatever you're doing. So control of all these mechanisms is done by the enteric nervous system and several hormones. That's what we're going to learn as we go through the process today. So this is the process. So when I was telling before about how start studying this stuff. So the sequence of events, first is chewing. So we have to masticate. You guys test for muscles of mastication, right? Clench your teeth, open your jaw, all that stuff is to make sure you actually can chew your food and move your mouth. It creates a bolus suitable for swallowing. If you want to lose weight, chew your food more. Take your time chewing your food. Don't just take 2 bites, chew it, and swallow it. If you break it up into smaller pieces, it's easier for your stomach to break them up even smaller from there. Also, remember we learned from sensory physiology. It takes some time for your brain to get the hormones, which we're going to learn today, that tells you, hey, you're full now, you can stop eating, the hunger goes away. If you take more time chewing than just swallowing, eventually your brain will be like, okay, fine, you're full now, you don't have to eat anymore. And you will eat less food because you're spending more time chewing your food. Saliva lubricates the food and provides some enzymes for digestion. So we have enzymes in our mouth that actually will turn starch to sugars. So giving kids crackers, thinking that, oh, it's not a sugary treat, you give them a cracker instead, well, they're going to turn that right into glucose in their mouth anyway. So that happens fast. So we chew the food. Now we swallow it, and it goes into the stomach for about 1 to 4 hours. And it starts mixing the food. That's all the stomach does, is it mixes your food. Okay? All potatoes that you eat are mashed potatoes in the stomach. It doesn't matter what they were beforehand. They're all mashed potatoes when they get down to the stomach. Okay? Stomach motility just grinds foods into smaller particles. It also increases surface area. If you have one piece of steak, 1 cube, you only have that surface area. But if you chew it up into smaller pieces, now you have bigger surface area for enzymes to get in there and break it down. Gastric acids assist in dissolving the food and denaturing the components of the food. So you all ate lunch. That food's going to sit in your stomach for the next 4 hours. If you had more liquid lunch, then it goes through faster because it doesn't need to be broken down as much. Food enters the small intestine with delivery of major exocrine secretions. So the pancreas, the gallbladder, those are exocrine. They're being secreted into a space. Pancreas secretes enzymes and bicarb. As we go through all these different parts of the body, we're going to talk about purpose of the structure. So when you talk about anatomy gives you the structure of everything. Physiology tells you the function of it. So your hand has a structure that allows you to squeeze an opposable thumb to do different things. That's the function of it. Bicarb. We have all this acidic chyme that's sitting in your stomach with all the gastric acid, so it's very, very acidic in your stomach. If you've ever vomited, you can feel how that burns coming up because all that acidic coming out. But when all this acidic foodstuffs leaves the stomach, what structure does it hit first? What's the first part of the small intestine? Duodenum. The duodenum in the small intestine is not built for acidity. So one of the first things that happens is pancreas dumps bicarb into the small intestine. Why is that important? What's it going to do? Neutralize it. That's what I'm saying. Think about what you're doing where the food is. The food's in the stomach. It's very acidic. We have all the gastric juices, the hydrochloric acid. That all stays there, mixed into the food. And when it leaves the stomach, all that acidic chyme is going to hit the duodenum. And when it does that, that stimulates a whole other slew of hormones that are going to be released. But the main one is this bicarb solution to neutralize that acidic chyme. If that doesn't work very well, you can get duodenal ulcers, where the ulcer will form in the duodenum because of all the acidity that's there now that shouldn't be there. Acid reflux. That acid in the stomach creeps up into the esophagus. You get a burning sensation. That's why people, when they complain about heart pain, if they do this, that's acid reflux. If they go like this, it hurts in this area right here, and they over an area, that's heart pain. But if they do this, it's usually esophageal pain. Gallbladder secretes bile, which is important for lipid assimilation. You ever try and mix oil and water? Does it work? No. What do you need in order to mix the two? An emulsifier. Who likes to cook? What is the emulsifier that we use in cooking? Egg. Egg will emulsify the water and the protein to allow it mixed together. That's how you get mayonnaise. If you're wondering how mayonnaise is made, that's what it is. The gallbladder secretes bile, which emulsifies the fat so that we can still absorb it. Okay, we have to get it into like a water-based molecule. So we have to emulsify it so that it can be mixed up. That's what the gallbladder does. Then the food's going to stay within the small intestine for about 7 to 10 hours as it goes through. There's like 30 yards, I think, of small intestine. It's a big amount of small intestine that's cramped in there. And all significant absorption of nutrients occurs in the small intestine. So when we talk about absorption, it does not happen in the stomach. All the stomach is a rock grinder. And it just mixes up the food into smaller and smaller pieces. That's all it does. When it gets to the small intestine, now we can start worrying about absorption of nutrients from the small intestine. Then once it gets its transit time through the small intestine, then we get to the large intestine, the cecum, sigmoid colon, finally the rectum, and that occurs about over 12 to 24 hours. So the food you eat today, you won't see it again until tomorrow when it does its whole trek through the body. Functions of the large intestine is mainly fluid and electrolytes. So a lot of the fluid absorption or even reabsorption in the large intestine of water happens in the large intestine, which is why a lot of the diarrhea stuff happens with the large intestine. Transit time through that, if it goes too fast, you get diarrhea. Too slow, you're constantly absorbing water. When it goes slow, and that feces gets very hard because you're extracting all the water out of it, and you get constipation. So elimination of fecal waste occurs about 1 to 3 days after eating a meal. That's the overview of the GI system. We eat the food, break it down, absorb the nutrients that have been made smaller and smaller, and we worry about water. There really is no homeostatic variable for the GI system. Just know that when you eat, we have to break these things down, smaller particles, and absorb them. So this is just kind of talking about the whole thing again in just one long tube with some accessory organs. Yes?
Do you even know the time frames that were mentioned in the last slide?
We'll go through them again. This is just an overview. We'll go through them again as we go through each section. Structural features of the GI tract. We know it's all smooth muscle, so we have to have a muscle layer. And we need to have ways to secrete and control of it. So there are 4 layers to the GI tract. The innermost layer that actually touches the chyme and then touches the food stuff is the mucosal layer. Okay, mucosa. It has endocrine cells that are going to secrete things into the bloodstream. It's got exocrine glands, which are going to secrete into the space itself, the hydrogen, the chloride, which combine to make your hydrochloric acid. They're going to be dumped as an exocrine gland. And we have mucus cells. We have to line the intestine with mucus. What does mucus do? Lubrication. Okay, that's why if you don't drink enough water over time, you start getting a little dehydrated and you start drinking things and you can feel it going down your esophagus because you're reduced the mucus layer. So you can actually feel it going down your esophagus. That's the mucosal layer. Does the mucosal layer have any blood supply in it? Do you see any? No. That's very important because when you talk about ulcers, if they're bleeding, we know we had to get into the layer that has the blood vessels because the mucosal layer has no blood vessels. Okay? So we have our submucosal layer, major blood and lymphatic vessels. So blood and lymph are here, and we also have neurons in this layer as well. So the blood supply is here, the nerves are here, and the lymphatic system are in this submucosal layer. So if you have an ulcer and it's bleeding, we know we had to erode the entire mucosal layer, and now we're starting to invade the submucosal layer because that's where the blood supply is. So submucosal is controlling the blood and the nerves in that layer. The next layer is the muscularis externa, and this is the major smooth muscle layer controlling movement through the GI system. Muscularis externa, and this has nerves as well. And the final layer is the serosa. So make sure you understand the order from most inside the lumen to outside. Mucosa, submucosa, muscularis externa, and the serosa. Those are the different layers of the GI system. So this goes into more detail. Mucosal layer, epithelium, highly folded to increase surface area. That's why we have all the villi. That has more surface area than that will. So by having these villi, these microvilli, it provides a great surface area for absorption. The exocrine secretions, mucus, electrolytes, water, digestive enzymes. Your endocrine cells release GI hormones into the blood. So anything that says endocrine means it has to go into the bloodstream. The next unit is all pretty much endocrine, with endocrine and reproductive physiology. Reproductive is all hormonal based as well, so the next unit is all just straight hormones. Cholecystokinin is one of them. We'll talk about the other ones as we get to the drawing. The submucosal layer, connective tissue, major blood lymphatic vessels, contains the submucosal or Meissner's nerve plexus. So the Meissner's plexus is in the submucosa. We have the muscularis externa layer, which is smooth muscle, and this is called the Auerbach nerve plexus. So the Auerbach nerve plexus is in the muscularis externa layer. The Meissner's nerve plexus is in the submucosal layer. And then the serosa is just the thin connective tissue on the outside. Everyone good so far on the different layers of the tract? So how do we control it? So it's a coordination of neural, hormonal, and paracrine mechanisms that all work together to allow you to break down your food. What can generate change in the GI system? Gut motility, epithelial secretion, and blood flow. There is a level of intrinsic activity, we call it GI tone, just like how your vessels have tone to them. Allow them to be a little bit constricted. The GI also has some sort of level of tone with them as well. Control of GI smooth muscle underlies gut motility. So if the gut motility is not working very well, the GI smooth muscle is not squeezing very well in those certain patterns to get the food moving, you're going to have slow motility. Most GI smooth muscle cells have slow waves, and that gives that rhythmic smooth muscle contraction. If you ever heard your stomach making noises or different weird things, it's just your normal GI motility kind of squeezing and moving things around. And voltage-gated calcium channels. So I'll go back to your lecture 1. We talked about smooth muscle, and it's that calcium calmodulin complex that they form because this is smooth muscle now. This is not skeletal or cardiac muscle. So there's a thing called a paralytic ileus. This usually happens after abdominal surgery, like the GI system kind of gets stunned from having surgery, and the whole GI system doesn't work. If the GI system is not working, what will you not hear on physical exam? Bowel sounds. Which is why after surgery, you don't let your patients eat or drink anything until they start, or not really drink, but more eating things, until they've either passed gas or have had a bowel movement. Because if they pass gas, that means their GI system is moving again and expelled out things, so then they can start eating again. But until any of those signs happen, you don't want them eating because the stomach is not really working very well because of the surgery. It's called the paralytic ileus, one of the major post-surgical complications. Okay, so the enteric nervous system, the gut-brain. How does the brain and the GI system kind of communicate with each other? It is a division of the autonomic nervous system. We did mention this a little bit before. PNS, SNS, and the enteric nervous system. I said wait till chapter 8. Well, we're here now, so we'll discuss it now. Mini brain, independent information processing capabilities. It has a program library. Okay, which is kind of why people get into habits, certain bowel habits, the same time every day they have to go to the bathroom because you kind of programmed yourself in a way, trained yourself through whatever reasons to only go to the bathroom at certain times of the day. Emetic activity is vomiting. So that has a program as well. That's called reverse peristalsis. Your body knows what to do if you eat anything bad to get it out of you. So responsible for moment-to-moment control of gut motility and secretion. So because we're talking about nerves, we have to talk about the layers that actually have the nerves. So the submucosal layer and the myenteric plexus. Those are the only two layers of the mucosal tract that actually have nerves in them. So let's see what they do. So the submucosal plexus, intestinal absorption and secretion via innervation of the epithelium, endocrine, and submucosal blood vessels. So secretion and absorption are controlled by the submucosal layer. The myenteric plexus is just gut motility, and these longitudinal and circular smooth muscles allows your GI system to kind of act like a snake. Instead of just squeezing, if you just squeeze in one area, the food won't go anywhere. But if you could squeeze it and turn, it's called rostral, I think, churning, it kind of twists, and that twisting like a snake-like, and it propels the foodstuffs to go in one direction. So it's just the way that the muscle layers are aligned and allows the type of churning and propulsion to keep the foodstuffs moving through the intestine. So make sure you understand those two layers, what the nerves are responsible for in those layers. We do have neurotransmitters here as well. Acetylcholine is the primary one. Secretion and motility. Acetylcholine, parasympathetic or sympathetic? Parasympathetic, which is what? Rest and digest. Now we're in the digest portion. So acetylcholine, secretion, and motility. Makes sense because those are activities that you would do during parasympathetic innervation, rest and digest. So acetylcholine helps with secretion and motility through the GI tract, therefore helping with digestion. ATP and nitric oxide are inhibitory in the GI tract. We also have these gut-brain peptides, and they're found in the enteric nervous system and the central nervous system. One of the big ones is vasoactive intestinal peptide, or VIP. What it does is a potent stimulator of intestinal fluid and electrolyte secretion, but it inhibits motility so that you have more time for digestion to occur. So it kind of slows things down so that we have time for absorption and digestion to occur. So when you start learning about drugs that you can give patients, if you give them an anticholinergic drug that's going to prevent acetylcholine from having its effect, one of them is amitriptyline, which is a tricyclic antidepressant called TCAs, if you ever heard of that. They inhibit the effect of acetylcholine systemically. So, GI side effects for an anticholinergic drug, dry mouth. Why? What does acetylcholine do in the GI system? Secretions, fluids and mucus. serous solutions, saliva. If you don't have that, you get dry mouth because you're not producing the secretions because acetylcholine is not working. Constipation, same reason. You're pulling out too much water. There's not enough lubrication in the large intestine, and it slows down gut motility. Constipation occurs. Ileus is not really working anymore. Nothing works. And nausea and vomiting because food is just sitting in the stomach. You keep eating more, eventually you'll get nauseous and vomit from eating too much food. So this is what happens with anticholinergic drugs. There's a whole bunch of other things with it, but we're in the GI system, and this is what those things do. So our gut-brain axis. Here is enteric nervous system. There is the central nervous system. We have our SNS efferents. So sympathetic nervous system inhibits intestinal motility. Does that make sense? Sympathetic is fight or flight. You don't want your stomach moving and digesting food because you're running away from a bear. PNS efferents, intestinal motility and exocrine secretion. You eat food, or you know it's like 11:30 and you're about to eat lunch. You start thinking about it. The parasympathetic will start kicking in to start getting your mouth watering so you're ready to start eating the food that you're going to have. Now, we also have afferents, which go from the stomach to the brain to kind of tell it what's going on. The SNS afferents give pain. So if you have abdominal pain, abdominal discomfort, it's your sympathetic nervous system that's delivering that pain signal to the brain, telling you that you have pain. Yes?
Why do, if some people, like when they're nervous, they have to use the bathroom?
We'll get to that very, very soon. It's coming up. PNS afferents. Okay, this is the parasympathetic system telling the brain what's going on in the GI system or in the enteric nervous system. Nausea, distension, satiety are all parasympathetic innervation or stimulus that goes to the brain. So if you have pain, sympathetic nervous system is telling the brain that you have the pain. Nausea, gastric distension, feeling of fullness is a parasympathetic trait. That's how the gut and the brain communicate. Any questions about this so far? So here's your thing. Emotions, anxiety can cause diarrhea as well. So some people, when they get very anxious, they get diarrhea. It's just, again, the brain and the gut brain talking to each other and bringing out that response. So that's when that happens. So they send these command signals, and during periods of anxiety, some people will get GI distress. It's just all part of the gut-brain connection. So parasympathetic innervation is your vagus nerve and the sacral nerve, S2 to S4. Those are the major ones for parasympathetic innervation. Efferent innervation generally causes excitation, more secretion, more motility. So this is under parasympathetic control. We want to get the foodstuffs moving through the system, and secretion makes it move easier. Vagus nerve is mainly just the upper GI, motility of the stomach and upper GI. It stimulates gastric secretion, pancreatic secretion, and contraction of the gallbladder. We need all three of these things to successfully digest and absorb your food. So they all have to be there. They're all under parasympathetic control, which makes sense. Rest and digest. We're going to break down our food. Afferent fibers, non-painful distension and nausea. Non-pain is parasympathetic. The pain is sympathetic. We have the vasovagal reflexes involve afferent and efferent signals to the vagus nerve. So distension of a stomach during meal stimulates your afferent signal, stimulates gastric acid secretion via the vagal efferents. So once you start eating, your stomach starts to expand because you're putting food in it. It's going to get bigger. When it starts to expand and distend, it sends an afferent signal to the brain for the parasympathetic to start getting gastric acid secretion going because we know we have to break down the food that's in our stomach. So just think about the food, where is it in your GI system, what's going on at that point. Sympathetic innervation, post-ganglionic fibers, we know from before, from doing the neurophysiology, sympathetic have a short pre, they go to the sympathetic side chain ganglia, and then they go out from there wherever they got to go. So it causes vasoconstriction. Okay, we don't want blood going through the GI system during fight or flight responses. Others will have a direct inhibitory response in the GI tract. Motility decreased, the tone of the sphincters are increased, so you don't want to have to do all those things. And remember, pain to the CNS is also sympathetic as well. Immune system, regulation in intestine includes our mast cells. So whenever you think of mast cells, think of histamine. They're the ones who release histamine. So if mast cells you start hearing mast cells, think histamine and what histamine is going to do. GI hormones, okay? We have hormones that are released by the stomach and they go into the bloodstream. Influenced by the chemical environment in the lumen of the GI tract. So if you have a lot of fat, high fatty meal, a lot of cholecystokinin is going to come out because that needs to get the gallbladder involved in order to emulsify all the fat you just ate. Make sense? So if you didn't have any fat in your meal, your gallbladder wouldn't really come out that much or be involved because you don't have a very high fatty meal. Enteroendocrine cells have microvilli that taste the gut lumen. So they're like taste receptors. They are going to see what food you just ate and act accordingly based off of that. Timing of the hormone secretion is guided by crosstalk with our enteric nervous system. And all these hormones have to go through the portal venous system before going into systemic circulation. This is called first pass metabolism. I think it's in this picture. No, it's coming up. When you take a pill, you eat food, the entire GI system drains into one vein called the portal vein, which then goes into the liver. So no matter what you eat, no matter where it's absorbed, they all get pulled into the portal vein first, and then they go into the liver, and then it goes to the body from there. The liver is going to metabolize whatever you're giving that patient. That's called first pass metabolism. So the quantity of drug that you give, you know it's going to go through the liver first, and the reduction may occur there. So that's why you have to give a little bit more of a drug than you want, because you know the bioavailability is going to go down because of the liver enzyme, first pass. If you give a drug intravenously, you bypass first pass metabolism because you're already giving it into a vein. It bypasses all that. So IV drugs don't go through first pass metabolism because they're not going through the GI system. So any pill that you swallow, food that you swallow, anything that you swallow, when it gets absorbed, they all collect in that portal vein to go to the liver first. And we'll get to that in a little bit as well. So we talked about this already. So all hepatic portal vein carries all nutrients, GI, hormones, absorbed drugs directly from the gut to the liver. Everything goes to the liver first. They clean it out. Whatever bad things are in there, the liver takes out and lets it go through. Okay, so let's go through the different hormones and how they're secreted from the body. So on the bottom left, when you see that purple arrow, that means secretion. The one line with the arrow is going to excite, and the one arrow with the line is going to inhibit. The green icon, little drop there, is going to be our gastric juices. The yellow one is bile. And the orange one is going to be pancreatic secretions. Okay, so when we draw them out, I'll use those colors. So let's look at the top part. The digestive tract hormones. We're going to talk about gastrin, secretin, cholecystokinin, and the last one, glucose dependent. So what does that mean? It needs glucose to be present. Insulinotropic. What is that going to do? Going to release insulin or inhibit insulin? Release it. Glucose needs insulin to get out of the bloodstream. So by eating foods that are high in glucose, we know our body knows that and starts getting insulin ready to go, so that when the glucose hits the bloodstream, your insulin is already elevated and ready to handle that glucose load. We have some other hormones we'll talk about a little bit later on, but motilin increases gastric motility. Easy to remember, motilin, gastric motility. Pancreatic polypeptide inhibits pancreatic secretions. Enteroglucagon stimulates glycogenolysis. What's that? Breakdown of glycogen into what? Glucose. And that gets dumped where? Into the blood. And glucagonogenesis. What are we making? Glucagon. We'll talk about that a little bit later on when we get to hormones. GLP-1 stimulates insulin secretion. That's why these drugs, these GLP-1 antagonists that they give out, Ozempic and everything, makes you not hungry because your insulin doesn't spike. It's supposed to be for diabetics, but they're using it too. Okay. And the four major hormones, the green is going to be gastrin. The orange is CCK, I think. Let me make sure about that. Yep. The purple X is GIP. And the blue square is secretin. So these are the four major hormones in the GI system. So, first hormone is gastrin. The stimulus for gastrin stomach distension. So you eat food, your stomach's going to expand because when you're in a fasted state, your stomach shrinks. And when you start eating, it starts to expand again. So when it expands, gastrin is secreted. The secretion occurs in the stomach mucosa. And the major action of gastrin: stimulate gastric acid production. We've eaten something, we got to get the gastric acid pumping so that we can break down that food, the smaller parts. So, gastrin, we said, occurs. When does gastrin come out? Stomach distension. So let's put a food bolus here, and it pushes out on the area around it. And when it does that, it causes secretion Okay, big wide arrow, so we're secreting gastrin into the bloodstream. Does that turn up okay? The green? Yeah? So food bolus goes into the stomach distends, the stomach mucosa secretes gastrin into the blood supply. This gastrin is then going to stimulate, okay, we just have the one arrow, so it's stimulating the gastric mucosa to secrete more gastric juices. And we have our gastric juices is in green, so put a red dot there, and we know that's our gastric juice. So that's all that gastrin does. Bolus in the stomach releases gastrin in the bloodstream, which then is going to tell the stomach, start releasing hydrochloric acid, the gastric juices into the stomach to digest our food. Start breaking it down. Okay? Everyone good on gastrin? Okay. Let's move on to secretin. What do you think secretin does? Secrete stuff. Okay? The stimulus for secretin is acidic chyme in the duodenum. What does that mean? That the food has left the stomach and now it's starting to go into the small intestine for further absorption to occur. So all that acidic chyme is coming into the duodenum. Now is when we start having to worry about secretin coming out to neutralize that acidic chyme. So the secretion occurs in the duodenal mucosa. So acidic chyme gets in the duodenum, causes secretin to be released. And what is the role of secretin going to be? Stimulate pancreatic juices. And we know that that is going to be high in bicarb. We have to neutralize the acid, so we need bicarb for that. Any questions about secretin? Okay, so let's talk about how this comes out. So we have our duodenum down here at the bottom. What's this structure right there called? Yep. Okay, so we get the acidic chyme coming in this area. Coming into the duodenum, which then causes the secretion of the other three hormones. Only gastrin comes from the stomach mucosa. All the other three are from the duodenum. So the secretin is, what color is we do for secretin? Blue, okay. Okay, so the secretin goes into the bloodstream, and it goes around and it travels till we get to the pancreas, which is not really drawn in, but I'll draw it in right there. There's our pancreas. So the blue travels around and it stimulates the pancreas to release the gastric juices, which are going to go all the way through there into the duodenum. What is that little structure down there called? Sphincter of Oddi. Okay. This sphincter of Oddi has the gallbladder, bile duct, and the pancreatic duct. Both connect to one tube, and that's controlled by the sphincter of Oddi. So, acidic chyme in the duodenum. Secretin comes out, stimulates the pancreas to make more pancreatic juices. It's an exocrine gland because we're secreting the actual substance into a tube, which is then going to the GI, into the small intestine, the duodenum. Any questions about secretin? Why is it out? Neutralize the acid. The next one is CCK, cholecystokinin. What does chole refer to? What does the cysto refer to? The space. Okay, so the cholecysto is the gallbladder. Kinin is going to be this thing that's going to stimulate it. So, stimulated by, let's blow this up, triglycerides, fatty acids, and amino acids in duodenum. Fatty meal, cholecystokinin is going to come out to help contract the gallbladder and provide the bile to come out. Secretion, duodenal mucosa again, and the stimulation positive gallbladder contraction, which allows bile to flow. Also, the sphincter of Oddi relaxes to allow the pancreatic and the bile to flow freely into the intestine. So you have to get rid of that sphincter, relax it so that everything can flow through. That's the role of cholecystokinin. The gallbladder itself, all it does is it stores and concentrates the bile. That's all it does. And when you have a fatty meal, cholecystokinin tells the gallbladder to contract, so the bile leaves the gallbladder, goes down the bile duct into the common one, and then into the intestine. The last hormone is our GIP. And the stimulation for this is glucose fatty acids and amino acids in the duodenum. So glucose is the big one. You had a meal high in glucose. Eventually, all that glucose that you ate is going to get into the bloodstream. GIP gets your insulin raised in your bloodstream, ready to go for that glucose you're about to eat. Diabetics will artificially give themselves the insulin based off of the amount of glucose they're eating in their food, because their pancreas isn't working to do that for them. So they take insulin to allow for all the glucose in their meal they just ate, enough insulin to correct all that glucose. Location of secretion, duodenal mucosa again. And the effect, positive pancreatic insulin secretion. So it's going to stimulate that, get the insulin going so that it's ready to handle all that sugar load. It also is going to inhibit gastric acid secretion. Why does that part make sense? Not the insulin. So think about the food you ate. If is now leaving the stomach and is going into the duodenum, is there any more food coming into the stomach? Theoretically, no. People may want to continue eating. But if you guys right now, you ate lunch an hour ago and now you're fasting essentially, there's no more food going into your stomach. So the GIP, if there's chyme in the duodenum, that means it's leaving the stomach. Do I need to continue making hydrochloric acid? No. This shuts it off because it signifies to the body, hey, we're done with the food in the stomach. We don't need gastric acid anymore. Let's stop producing it. Because what's going to happen if you keep making gastric acid with no food in there? Ulcers, acid reflux, all those weird things are happening because you don't have enough food in there to kind of like absorb that acid, and it keeps building up. So, I didn't do the cholecystokinin either, so I'm going to do that real fast. That is orange. Okay? So, we know the cholecystokinin goes into the bloodstream. It's going to go up to stimulate the gallbladder, right? What also does cholecystokinin do? Relaxes the sphincter of Oddi. So we're going to do another arrow. Here comes our cholecystokinin. We're flowing around. Now we're going to go up and we're going to inhibit. Can you guys see that? I know orange kind of blended in there, but the cholecystokinin is going to inhibit the sphincter of Oddi. Therefore, relaxing it to open to allow the bile and the pancreatic enzymes to get into the duodenum. If they don't relax, that sphincter is going to be tight and nothing's going to happen. So that's the cholecystokinin. The last one is the secretin. So it's the purple X in the bloodstream. And this purple X does a couple of things. Travels in the bloodstream. When it gets to the pancreas, what's it going to do to the pancreas? Stimulate it or inhibit? Stimulate what? Insulin. Now the pancreas has two functions, an exocrine and an endocrine. We're learning about the exocrine functions in this lecture for today. How does the pancreas help in digesting your food? Secretin, bicarb. We're going to talk next week about the endocrine pancreas when it comes to insulin and glucagon and somatostatin and other things. That's next week, yes?
Is this in chronological order? Like each hormone being released?
It kind of happens, they all at the same time. Once the duodenum hits, the acidic chyme hits the duodenum, all three hormones get released. Some may in more quantities depending on the makeup of the food. But again, if you have very high fatty, a lot more cholecystokinin will come out. High glucose, more GIP comes out. What also does the GIP do? So, so this X keeps going up. We get to the stomach. Now it's going to inhibit gastric acid. So those are how the four major hormones have their effect for digesting the food. What's the first one that's going to come out? Always. Why gastrin? What stimulates it? Distension of the stomach. That's going to happen first before the duodenum gets distended. So gastrin has to be the first one always. Then once the chyme hits the duodenum, then the other three come out. Okay? Any questions about these four hormones? Make sure you know all the four hormones, what they're doing, what stimulates it. But again, if you just think, I'm putting food in this area, what has to happen? Just think about it that way. In each section of the GI system as we go through it, what's happening where? Okay, I think I have like two things. What's the last one we're done? We also have paracrine control. What is paracrine? Exocrine is into a space. Endocrine is into blood. What is paracrine? Next to it. Whatever's next, cells next to it. It's going to affect the cells right next to it. So we have serotonin produced by enterochromaffin cells in the intestinal mucus in response to distension of the gut wall. So you eat food, serotonin comes out. That's why some people feel better when they eat food, because food can make you feel good. Effects are generally excitatory, increase intestinal motility and secretion, and that's also a link, they think, between anxiety and GI issues is the serotonin as well. Somatostatin. Whenever you see this word, I always want you to associate stop with it. Somatostatin is going to mean stop. Somatostatins stop things. They're inhibitory. So whenever you see the word somatostatin, it's inhibitory, produced by D cells, potent inhibitor in the GI system. Stops pancreatic and gastric secretions, relaxes stomach and gallbladder, decreases nutrient absorption in the small intestine, so it stops everything. Potent vasoconstrictor as well. So somatostatin analogs like octreotide reduce bleeding from ruptured esophageal varices, because if you also give it for postpartum women when the uterus can't stop bleeding. Usually after delivery of the baby, the uterus starts to constrict real fast to stop the bleeding. Sometimes it doesn't happen as fast as you want, so you give this drug, which causes potent vasoconstriction, so all those uterine vessels clamp off and there's no more bleeding. That's what octreotide does. The last one is our histamine. These are made by enterochromaffin-like cells. So enterochromaffin cells make serotonin. Enterochromaffin-like cells, or ECL, makes histamine. Histamine has a stimulatory effect on acid secretion in the stomach. So if you have very high histamine levels, gastric acid levels go up as well. Cimetidine is an H2 blocker. It blocks the histamine. Volume and acidity of the gastric juices are reduced. This is how one of them works. The H2 blocker blocks the histamine channel. The other one we learned about a while ago, we talked about, was those proton pump inhibitors, which we'll talk about in a little bit. So let's take a break. We'll start worrying about the structures. Come back at 1:35. Okay, we'll get started. We'll get started. So this last part is just we go from top to bottom. We go through each section of the GI tract and kind of talk about the function of that area and what it's doing. So the first part is just mouth and esophagus. Mastication, we do have a chewing reflex. So once you start, the bolus hits the mouth, you start chewing. Lower jaw initiates the stretch reflex. So as you open your mouth, it's going to stretch reflex and have it come back up. And that's the chewing rebound contraction. Process is repeated until obviously you break it down into small enough particles and then you swallow it. Benefits: smaller food particles, increased surface area, more area for enzymes to work, prevents excoriation. What's that? Scratches. You don't chew a potato chip very, very well, and that little shard drags the whole way down your esophagus. You'll feel that. So chewing it helps. And helps break down indigestible cellulose membranes in fruits and vegetables. So like the outside part of a corn, we can't break that down, but by chewing it, we can get everything inside out of it and break it up enough so we can break down the stuff on the outside. Same thing with fruits and vegetables. They have cell walls, cellulose, which we can't break down. So we have to chew it to get it going before you swallow it. We do have a lot of salivary glands. We have primary secretions, which they can be serous, which is very, very watery, contains ptyalin. That ptyalin digests starches. Like I said before, you give a kid a cracker, they're going to break that starch and turn it into a glucose, a sugar molecule in their mouth. So they'll still get the sugar. And we have mucus secretions. Serous are more watery, mucus are going to be thicker. The mucus contain mucin for lubricating the food so that it can slide down the esophagus easier. So we have to lubricate it as well. We have the glands here. The parotid gland is mainly serous. Submandibular or sublingual are serous and mucus. And we have our buccal glands, which are mostly mucus secretions. Daily, about 0.5 to 1.5 liters a day of saliva your body produces just in the mouth. We have to reabsorb all that water so we don't get dehydrated. Function of saliva also helps with oral hygiene. A lot of pathogenic bacteria in the mouth. That's why they say like a human bite is probably more dangerous than a dog bite because we have all this extra stuff in there. But what does saliva help do? Wash away bacteria and food particles. Contains thiocyanate and proteolytic enzymes, and also has those antibodies as well. Which antibody are we talking about? Is IgA. Okay. Swallowing, deglutition, raises a soft palate and swallows. Again, these are all the nerves involved with swallowing. You don't need to know all of them, memorize them. But obviously, there's a lot of cranial nerves involved with the process of swallowing, which is again why anyone coming in with any type of weakness, neurological complaint, don't give them any food or a stroke until you've ruled out stroke and that they can swallow properly. Deglutition is swallowing. There are 3 phases to swallowing. The first one is the oral or the buccal phase, and this is voluntary. This is you actually chewing up your food and starting the process of swallowing. Voluntarily squeezed and rolled posteriorly into the pharynx, and the tongue presses up against the palate, and that pushes the food back. The next phase is the pharyngeal phase, and this is involuntary. This is initiated by the swallowing receptors, posterior cavity, and oral pharynx. These are just the processes that happen so you don't swallow your food in the windpipe. So as you swallow, the uvula soft palate lifts to cover the posterior nares so that the fluid doesn't go up to the nose. We want all the food to go into the stomach, not up the nose. We have palatopharyngeal folds pulled medially to approximate each other. This prevents large materials from passing through. Vocal cords approximate. Larynx is pulled upwards anteriorly. Epiglottis swings back and covers the vocal cords and the trachea. So you don't swallow and aspirate the food. And finally, the upper esophageal sphincter relaxes. That is a very important one. We'll talk about that a lot when it comes to GERD in a little bit. And wave of peristalsis propels all your food straight down to your stomach. You could be upside down, swallow something, it'll still go to your stomach, go up against gravity. The peristaltic waves will take care of all that for you. So that's deglutition. The final phase is the esophageal phase. Again, swallowing center of the brainstem. This is under vagus control. We have primary peristalsis, which is continuation of the pharyngeal peristaltic wave. Passes from the pharynx to the stomach in about 8 to 10 seconds, 5 to 8 if upright. So if you swallow something, it takes about 5 seconds to get from your mouth to your stomach. Secondary peristalsis, any residual food in the esophagus, intrinsic neural circuits into the myenteric nervous system. Why the myenteric? What layer is that in? Muscularis layer. That provides all the squeezing and the peristalsis. You need that layer to work. And then we have receptive relaxation in the stomach. So when that food hits that first part of the stomach, there is a reaction or reflex where the stomach will start to relax and to distend because it's thinking that you're going to have a meal and it's going to get ready for it. So that's the final esophageal phase. Finally, get to the esophagus. It's only about 10 inches long, passes through the diaphragm via the esophageal hiatus. That's where you get a hiatal hernia. The stomach pops up through that opening and it gets caught, or it could get caught. We do have esophageal secretions here because it's basically making a slip and slide. That's all you have to do. Got to get from the top to bottom, got to get it lubricated as possible so that food can slide down. The lower esophageal sphincter opens to allow food to pass into the stomach, and normal situation stays closed to prevent regurgitation or reflux, that acid to leave. So that lower esophageal sphincter, the tone of that sphincter is very important when it comes to like acid reflux. So GERD, when the lower esophageal sphincter is incompetent, doesn't squeeze all the way, and some acid can squeeze through. Factors that predispose to GERD: smoking, obesity, pregnancy, hiatal hernia. These are all things that are going to give you that heartburn sensation. These things weaken the lower esophageal sphincter, and if it weakens it, you're more susceptible to like acid reflux. That's it on the mouth and the esophagus. Just grinds the food to smaller particles, transports it down to the stomach. So now we're in the stomach. There are 5 major anatomical areas for the stomach: the cardia, fundus, the body, gastric antrum, and finally that pyloric sphincter to empty the stomach into the duodenum. The cardia, fundus, and the body comprise the ozintic or the parietal layer or area. Major exocrine secretions from this area, the parietal layer. So we'll talk about the parietal area pretty soon. The pyloric gland is the distal 20% and a major source of gastric hormones. Stomach mucosa has gastric pits and gastric glands as well, which are going to secrete stuff. So this is the major part right here. This is the gastric pit, and you can see how you go into these pits. There are different types of cells that line these pits. We have the ozintic parietal cells, and they're in the dark brown ones. You'll see it point out. And what do they secrete? HCl and intrinsic factor. What's intrinsic factor for? B12 absorption. And where does that occur? The B12 absorption from this intrinsic factor. This is in the terminal ileum, right before you get to the large intestine. So if anyone has a gastric bypass surgery, you have to ask them what kind of surgery do they have. Some of them bypass the terminal ileum, so what are they going to have? Or potentially could have? Yep, all those problems with anemia is because they're not getting the B12 back, the pernicious anemia. Peptic or chief cells secrete pepsinogen. Because it has the ogen at the end, what does it mean? These are inactive. And we'll talk about when this becomes active later on. But pepsinogen comes from the chief cells. The D cells are your somatostatin, and the G cells are gastrin. So you can see the G cells are mainly at the bottom. Parietal cells are getting closer to the top. Here's your mucus cells. They secrete a lot of mucus to help line the stomach. Yes?
Is this where the rugae are in the stomach?
Yes. Those are those folds. Now, here's another picture. This is talking about the parietal cell. What do the parietal cells secrete? hydrochloric acid, and intrinsic factor. So this is the entire parietal cell. Let's talk about things that stimulate the cell to make more hydrochloric acid, more gastric juices. Here's one, acetylcholine. Does that make sense why that would increase gastric acid production? Acetylcholine, parasympathetic, rest and digest. Let's get things going. Acetylcholine gets more hydrochloric acid secreted. Here is your histamine from the enterochromaffin-like cell. Histamine by itself is stimulatory to the parietal cell to get more gastric acid secreted. This is where you have those H2 blockers. They block the histamine from hitting these cells. If you block the histamine, what are you going to reduce? Gastric acid secretion, because you're reducing the histamine. The gastrin that comes from the G cell, remember it goes into the bloodstream. The gastrin is the only one secreted by the stomach mucosa. The other three are all duodenal. So gastrin has to come from this area. And what gastrin does is it stimulates the enterochromaffin-like cell, which is what it's going to do what? So when gastrin stimulates the ECL cell, what is that going to do? Increase histamine, which is going to increase what? Gastric juices. Gastrin also by itself is stimulatory to the parietal cell to make more hydrochloric acid as well. So the gastrin has two effects, a direct effect on the cell and an indirect effect by stimulating the ECL to make more histamine. On the other side, we have our D cell. D cell makes somatostatin. What did I tell you remember about somatostatin? Stops things. So somatostatin is going to stop, put it up there, stop the parietal cell from making hydrochloric acid. And doubly, it's also going to inhibit the ECL cell from making histamine. So somatostatin, just like gastrin, has two ways of stimulating HCl gastric juices. Somatostatin has two ways to stop it. Stops it directly at the parietal cell and inhibits histamine. Both of those are going to reduce gastric acid production. So if you have a gastrin-producing tumor that produces a ton of gastrin, what is going to happen to your hydrochloric acid? It's going to increase because you have a ton of gastrin being produced, and we know what gastrin can do. Okay. Gastritis is just inflammation of the gastric mucosa. This will not have any bleeding. Why? What layer are we only dealing with? Just the mucosal layer. Is there any blood supply in the mucosal layer? No, therefore no bleeding. Just get some irritation. Causes smoking, alcohol, NSAIDs, chronic stress will cause some gastritis, some inflammation of the lining. So, overview of gastric functions. What's the stomach going to do? Motor function. Okay, reservoir for our ingested food, and then it just mixes and grinds it. It's like a rock tumbler. Just gets it into smaller parts. The exocrine functions of the stomach. Okay, it's going to dump some water into there to help dissolve and dilute the food. So water gets dumped into your stomach. Acid via HCl denatures the proteins. Also kills any microorganisms. Okay, having a low pH will kill the majority of the bacteria and things that get into our food. It's when there's too much of the bacteria or the virus or whatever, then the HCl can't take care of all of it, and some of it will get through, causing problems. We have enzymes that get dumped into there for pepsin, gastric lipase. They're for protein and fat digestion. Here's our intrinsic factor for B12 absorption. And our final thing, which is very important, the mucus bicarbonate barrier. This prevents the stomach from eating itself. This is acid. And I mentioned before when we first started talking about this unit, there are certain cell types which are resistant to acid. They're in the stomach to prevent you from digesting yourself. The important is this mucus bicarb barrier. Mucus provides a thicker area for diffusion and protects the layer. The bicarb, why is that important? It neutralizes the acid. So if any of that acid tries getting close to the stomach lining, that barrier with the bicarb is going to neutralize it to protect the lining of the stomach. When that mucus bicarb barrier starts to erode and go away, then you start getting your defense mechanism weakens, and you can start getting ulcers and some other problems. The endocrine function, gastrin, stomatostatin, and ghrelin. Ghrelin is the hormone that tells you that you're full. So when that ghrelin comes out of the stomach, it goes to the brain and it's like, hey, we've eaten now, we're okay, you can stop eating, you're full. It's the ghrelin that tells your brain that you're full. Gastric motility, so our stomach's got to move too. So in a fasting state, nothing going on, every 90 minutes, you get a migrating motor complex. So if you haven't eaten for a long period of time and you hear your stomach making noises, it's usually going to happen every 90 minutes, and it's just a stomach getting smaller and smaller. Food enters the stomach, we get what's called the receptive relaxation. Food enters the stomach, we get a relaxation of the stomach, and it gets bigger because we are anticipating you're going to be eating a lot of food. Next, we have what's called ingestion, where the proximal stomach slowly sustains tonic contractions, press food into the distal stomach. So we kind of squeeze it in the bottom and squeeze back up. And it gets squeezed back and forth in the stomach and gets churned around. Again, mixing with all these enzymes, mixing with the hydrochloric acid, getting these things made smaller and smaller, denature the protein so that eventually when we get to the small intestine, we can absorb all this stuff. And finally, we have antral systole, food mixed with gastric juices to reduce the particle size. So, mechanism of gastric acid secretion. We got to figure out how we can get hydrogen and chloride both to be secreted into the stomach. So there's two ways to do it. We got to get the hydrogen out and we got to get the chloride out. Let's deal with the chloride. Where does that look familiar? Where have we seen that before? Chloride shift, right? Red blood cells trying to get that carbon dioxide to mix in and so we can transport it everywhere. So we have that here as well. Chloride comes in. This is the gastric lumen. And we have the interstitial space, and this is the wall of the cells. I have all the cells in it. So this alkaline slide or chloride shift brings the chloride in, which then goes through that channel to get the chloride in there. That's all that does with the chloride. Now, how do we handle the hydrogen and the potassium, or the hydrogen really? This same bicarb that came out of the cell is being created inside the cell via this carbonic anhydrase. Okay, so the CO2 comes in through carbonic anhydrase, is going to produce the bicarb and the hydrogen. So CO2 combines with water via carbonic anhydrase gives you the hydrogen plus the bicarb. Right? Now, we're left with the hydrogen and the bicarb. That bicarb will contribute to the alkaline shift, that chloride shift. That bicarb just leaves and brings chloride in. That hydrogen, via the hydrogen-potassium exchanger, that hydrogen that's created from the CO2 now leaves. We bring the potassium back in. This just shows how we get rid of the potassium. So we're always maintaining our normal potassiums. But look what we've created inside the lumen. We brought the hydrogen in, and we brought the chloride in. They combine to make the hydrochloric acid, which is our gastric juice. That's how we get hydrogen and chloride into the lumen of the stomach. to make the acid. Okay, the hydrogen comes from the carbonic anhydrase activity. The bicarb that's created from that leaves to bring in the chloride in. And that's how we get hydrochloric acid. Okay? So proton pump inhibitors are another way to reduce gastric acid, such as omeprazole. And what they do, they inhibit that hydrogen potassium ATPase pump. If you inhibit that, is hydrogen going to get into the stomach? No. Therefore, you reduce hydrochloric acid. So that's how PPIs work, proton pump inhibitors. They're blocking the proton pump, which is this. And they block it. So hydrogen can't get in, you can't make can't make hydrochloric acid. Whereas the H2 blockers block the histamine from acting on the gastric cell to make more hydrochloric acid. This does it this way, yes?
Is that for something like GERD?
Yep, they both are. Okay, stimulation of gastric acid production. Obviously, vagus nerve, acetylcholine. We get endocrine from gastrin, paracrine from histamine. Now, prostaglandin E2 is an antagonist of histamine by inhibiting production of cyclic AMP. Don't worry about that right now. We'll get to that more in endo when we come to secondary messengers. But, so prostaglandin E2, is this protective for you? What is it doing? Does it increase stomach acid or reduce stomach acid? So it's protective. So prostaglandin E2s are good to have. They help with that mucus bicarb barrier. So NSAIDs inhibit prostaglandin formation. So it reduces our mucus bicarb barrier, making it easier for acid to hit the wall. That's why people who take ibuprofen for long periods of time, Motrin, like 4 times a day, every day, over time we'll start getting some acid problems, some like gastritis or something, because the prolonged use of NSAIDs reduce the amount of prostaglandin E2, reduces the mucus barrier, which is your defense, so you make it more easy for acid to hit the stomach. So that's what prostaglandin E2 is. This is when we talk about, again, H2 receptor antagonists sold over the counter as antacid agents. This is where your question is, Alexa. So these are proton pump inhibitors are more effective because they block the HK ATPase pump, whereas H2 receptor blockers just block histamine from working. Both reduce gastric acid. So this is just a review talking about different phases of the stomach. Cephalic phase, this is, you're at Chili's and the waiter's walking across with the fajita plate. You smell it, it's not yours, but it's like, oh, where's my food coming? That starts the phase, your mouth starts watering, that's the first phase of the stomach. Cephalic phase, so sight, smell, taste of food. The gastric phase, this is the distension of the stomach. So as food is now in the stomach distends. That's the next phase in the gastric phase. And then finally the intestinal phase. And this is obviously getting it out of the stomach into the intestine. Once it's there, we don't need the stomach anymore. So the stomach starts to turn off hydrochloric acid because we don't need it anymore. Food has left the stomach. So like I said, when you're studying this, start thinking you're a piece of food. I'm in the mouth. What's going to happen in the mouth? I swallow it. What are the phases of swallowing? The 3 phases. Now we're in the esophagus. What's happening there? Follow that food down. Now we're in the stomach. What's happening in the stomach to get the hydrochloric acid out? Blah, 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 as you go through this. So here are the pepsins. Pepsins are proteolytic enzymes that attack the peptide bonds and make them into smaller amino acids. We said before, they are released as pepsinogen, which is the inactive form. There are 2 types. You don't got to worry about where they come from. The important thing is this. Pepsinogen becomes pepsin when the pH is less than 5. Is the pH less than 5 normally in the stomach, do you think? Normally it's not, it's a little bit higher, around 5, maybe a little bit higher than that. We don't need pepsin active because we don't have any food in there. When you start eating, what are we going to produce a lot of in the stomach? Gastric acid. What's going to happen to the pH? It's going to drop because we ate something. Now the pH drops. It's going to activate all that pepsinogen into pepsin, which will start breaking down your proteins. But it only happens when the pH is low, meaning there's food in the stomach. Otherwise, the pH comes back up because you're not eating. There's no reason for the hydrochloric acid. And the pepsinogen stays as pepsinogen. We don't need it as pepsin anymore. That's where Pepsi Cola came from. It was originally marketed as a dyspepsia drink for like gas. So they called it Pepsi. It's supposed to be a dyspepsia. That's where Pepsi came from. And stimulated, again, ACH. Okay, makes sense. This is a parasympathetic process. Gastric mucosal protection prevents erosion due to the acid and neutralizes the acid because of the bicarb that is in that mucosal layer. So erosive gastritis can occur. This is chronic NSAID use I talked about before as a clinical correlation. These prostaglandins help with that bicarb barrier, and NSAIDs reduce the production of these prostaglandins. Therefore, your mucosal barrier defense weakens, making it easier to get ulcers, gastritis, and so forth. So chronic NSAID use is bad. Now we're moving on to our small intestine. First 10 inches is the duodenum, the middle 2/5 is the jejunum, and the final last 3/5 is your ileum. Secretions from all your salivary glands, okay? Remember that leader that you produce in the mouth? Stomach juices, pancreatic juices, liver juices, all mixed in the small intestine for digestion and absorption. Surface area increased via villi in the enterocytes, mucus-secreting goblet cells. Whenever you see goblet cells, think of mucus. They're the ones who create the mucus that helps the lining, and our endocrine cells. So you can see here, we have a crypt down at the bottom. We have our villus tip. These villi have microvilli, little brushes that further increase surface area. So all the absorption occurs up at the top. The new cells start at the bottom. So these are new cells. These are old cells. Cells are forced upward as they create a new lining. So all the new cells are at the bottom. The top of the villi are the old cells. This is what you rip away when you have like lactose intolerance, celiac disease, those gluten sensitivities, the brush border that you're wiping out. This is what we're talking about here. And so this is what I'm talking about. Celiac sprue is a malabsorption syndrome, hypersensitivity, resulting in destruction of the small intestinal villi. Diarrhea, steatorrhea, what's that? Fat in the feces is steatorrhea. Why would there be fat in the feces? What's not working well if that happens? Gallbladder. Abdominal bloating and some flatulence. Removing gluten will resolve the condition, but takes a few days because the crypts require more days to mature. So it takes some time to rebuild it. Fluid absorption. Typically, the jejunum will get about 7 to 10 liters a day. Small intestine will reabsorb 6 to 8 liters a day just by osmotic gradient. The reabsorption of fluid is not subject to the RAS system. The RAS system, the renin, angiotensin, aldosterone system, only affects the kidneys. Doesn't affect the GI system as far as its absorption of water. Separate process. Small intestine has very high epithelial water permeability, which is why if you just drink things, it gets absorbed faster. Because the water gets absorbed faster. That's why if you drink water by itself, you'll rehydrate yourself faster because the water doesn't need to be digested or broken down. It's already in the liquid and you just absorb it right away. Like protein powder for mixes, they're already ready to go for absorption. So that's why protein powder is good, because you just put it in the water, drink it, and you can absorb it faster because it's already been in its smallest unit to be absorbed faster. If food has very high water content and hypotonic to the plasma, we'll get rapid water uptake. So free water by itself, is it hypotonic to plasma? What's plasma concentration? 0.9% sodium chloride, right? 0.9% tonicity. If I'm drinking water, what's the tonicity of the water I'm drinking? 0. So I have 0 in the stomach. I have a lot of solute in the blood. Water will go right into the system faster because osmosis goes, it's faster. If the meal is hypertonic and water will initially enter the small intestine. So if you have a very hypertonic solution, very, very, very salty meal, your body is going to dump water into the small intestine to dilute that out. What will that cause to your blood volume? Decrease, which will cause you to experience what? Dehydration. And that's why you get thirsty when you have very, very, very high salty foods. Because your body's going to have to pull water into the small intestine to dilute out all that hypertonicity. And we get fluid absorption, relies on active transport of nutrients and electrolytes. Finally, at the very end, the colon will absorb 1.9 of the 2 liters of fluid delivered to the large intestine. And so 0.1 liters of fluid is still in the feces when you go to the bathroom. Still a little bit left, but most of it has been reabsorbed by the body for the water. So here's our duodenum. There's the ileum. So we're going downstream in the small intestine. Here is our luminal membrane. Here's the basolateral membrane, and this is going to the blood. You can see here sodium is tied to most everything to get absorbed. All of our nutrients need sodium there to get across. Our chloride, obviously this is just salt in general, both of them will go together. Sodium and hydrogen go in opposite directions. Again, remember, our blood is naturally going to be acidic. We want to get rid of that acidity. We can dump it into the feces. We can dump it into the urine. That's why a lot of the waters, if you look at all the different bottled water, some of them have different pHs. You want to drink the ones that are alkaline. Okay, that's why, who was it? The girl that does a stupid goop. Gwyneth Paltrow. Okay, so here's an example. She's like, drink alkaline water, which is great. But what does she add to it? A lemon. What's the lemon going to do to your alkaline water? Neutralize it so you're drinking plain water. I don't get where these people say, think about it. If stuff's going into your stomach doesn't care where it came from. All it knows is fat, it's carbohydrate, it's protein. The problem is all this ultra-processed stuff. So anyway, sodium has to be there for our nutrients to be absorbed. So that's why sodium is very important. We don't want to reduce our sodium to nothing because we need it for absorption. Small intestine also has secretions. We have these Brunner's glands, and they're in the first couple centimeters of the duodenum. And their job, large amounts of alkaline mucus. Why is that important right in that first little bit of the duodenum? All that acidic chyme from the stomach is going to go right there first. So it makes sense these Brunner's glands have a lot of bicarb there, ready to neutralize any acidity that comes in there. And it's stimulated by acetylcholine. Makes sense. Acetylcholine is parasympathetic, rest and digest. Secretin. Why does that make sense? What does secretin do? Increase more bicarb secretions from the pancreas, and obviously it's going to help with these things. Duodenal irritation. What is that duodenal irritation caused by? The acidic chyme coming in. The cell type in the duodenum is not made to handle low pHs. The stomach is. The esophagus is not, which is why when the acidity gets into the esophagus, you get heartburn and it hurts, because those cells are not made for it. So those are the Brunner's glands. Bicarb. First little bit of the duodenum, neutralize that acidic chyme that comes in. The next ones are these crypts of Lieberkühn. And they're over the entire small intestine. They have a couple of different cells. Goblet cells, mucus. Provide the lining, keep it nice and slick so that food can pass through easily. Okay, you want it to be like a slip and slide. You want everything to be mucus, slippery, so that food can move around. When it gets dried out, food can't move anymore. Then we also have enterocytes, and they're for water and electrolytes. Usually going to be absorbed pretty fast. But those are the two different types of cells. Okay, so Brunner's glands are the bicarb. Crypts of Lieberkühn are more mucus than water, kind of keep it slick and lubricated. Okay, those are two different types of cells we have in the small intestine. Fluid secretions, again, crypt cells needed for lubrication. Also, fluid secretion provides a source of sodium. Okay? If you don't have enough salt in your food and amino acid uptake, the body will dump salt into the intestine just to help with absorption, because all those nutrients need sodium there in that co-transporter to move into the blood. Mechanism is very important because it can be activated by bacterial enterotoxins that cause secretory diarrhea. Okay, if you ever been to like different countries where they have different flora and fauna in their food and everything, and you eat those things, you get stomach problems. It's because you're not used to those type of things that are in the food the way they are. It can cause these enterotoxins that cause huge amounts of diarrhea to occur. Water secretion occurs via osmosis. The key step is opening of the chloride channels in the luminal cell membrane. So we have two different types of chloride channels in our luminal membranes. One is a cyclic AMP activated chloride channel. The other one is a calcium activated chloride channel. Okay. The cyclic AMP is where VIP has its effect. The calcium channels are from serotonin and acetylcholine. Those affect the cyclic, the calcium activated chloride channels. Where this becomes important is, I think I threw out that slide. Basically, just understand that we have chloride that has to happen. A bacteria, the enterotoxin, can turn on the cyclic AMP chloride channel and start dumping chloride into the intestine. When that happens, water has to go with it like crazy, and you get diarrhea. So that's kind of where you'll see later on where these enterotoxins have their effect. Digestion and absorption, we have to break it down via hydrolysis into the smaller and smaller substrates. So proteins got to be broken down to amino acids, so then therefore they're small enough where we can absorb them. Carbohydrates need to be broken down to the monosaccharide version. Sucrose, glucose, fructose, lactose, those are the monosaccharide versions. Broken down into those smaller particles, the monosaccharide, which we can then absorb. Here we have our fat globules need to be combined with the bile to emulsify it. So we have our emulsified droplets, which then can be turned into free fatty acids, which we can then absorb. The free fatty acids are small enough. So that's, digestion and absorption. Vitamin absorption. We can't really synthesize a lot of these, but the body requires them for metabolism. So we have to talk about vitamins. Vitamins A, D, E, and K are your fat-soluble vitamins. Okay, absorbed by simple diffusion because they're fat-soluble. They don't require anything else. And they appear in those chylomicrons, which are just smaller fat molecules. Vitamin B, C, folate, biotin are water-soluble. Absorption is slow, and we're trying to figure out other ways to get those absorptions to happen faster. So remember, B, C are your water-soluble ones. All the other vitamins are fat-soluble. These just talk about what deficiencies you may have with certain vitamins. You don't have to know this one. But one of the big ones was scurvy with pirates and the people that go across the seas, those transatlantic trips. They weren't bringing any fruit with them, so they had no vitamin C, so they started getting scurvy. That's why a lot of they all have like lime or lemons or oranges and a lot of the pirate stuff because they needed the vitamin C, and they would take those things and have to eat them so they would get their vitamins. Vitamin A, pigment in retina, so you can get night blindness if you have deficient in vitamin A. Here's your B2 is riboflavin, that's your normocytic anemia. So you don't have to memorize this one, just showing you, yes, we have to have vitamins, we have to be able to absorb them and use them. That's it on the small intestine. Any questions on that? We need sodium to absorb all the nutrients. Yes?
Why is it bad then to drink lemon water or not?
Bad to drink lemon water?
Yeah.
No,
Not bad.
that's not bad.
Okay.
It's fine. Supposedly there's chemicals in the lemon that can survive, and they help with like capillaries. If people get a lot of bruising, easy bruising, just add just drink a cup of like a teaspoon of like lemon juice, and then that helps with like the bruising and everything like that. But lemon water, it helps. Like it helps with certain things, but it's not going to do anything miraculously to you. Okay, large intestine. The main function of the large intestine at this point is just water, electrolytes, vitamin K, and some of the B vitamins for absorption. The main one is going to be water and electrolytes for the large intestine. Production of vitamin K and B is via organisms in our gut. These little bacteria, they live in the large intestine, and they eat the stuff that we provide them, and they give us vitamin K that we then absorb. So without these flora in our gut microbiome, we can't produce vitamin K. Have a lot of problems that way. And the large intestine also has a storage of feces. Several hundred different species of bacteria, we call it a microflora. Some are commensal, meaning the bacteria benefit, and we aren't harmed or benefited from it, but they live there. Some are mutualistic, both bacteria and we benefit. And the bacteria are going to be mostly anaerobic because there's not a lot of air in the large intestine, so they're going to be mainly anaerobic bacteria. So microbes make vitamin K and some of our B vitamins. They can make fatty acids from cellulose, and some are used for energy. They help absorb our electrolytes as well. It outcompetes harmful species of bacteria. So when you take antibiotics, the antibiotic that you're taking doesn't care where the bacteria is located. It's just going to target those specific bacteria. So when people are on antibiotics for a long period of time, it can wash out the flora in your large intestine as well. So we give probiotics when you take certain antibiotics. One of the major problems that comes with this chronic antibiotic use, because you're getting rid of all the bacteria, the good and the bad, opportunistic bacteria can start to flourish. One of the major ones is C. difficile, Clostridium difficile. So if you're working in a hospital and you've heard of people who have C. diff, it's because all of you right now have C. diff in your intestine. But we keep it at check, we keep it at bay, and it doesn't cause any problems. But if you wipe out all the bacteria and the C. diff is left alone, it's got all this party going on, all this food to eat, it's going to go rampant in overgrowth. And you get another problem with that. But as an example, we have these bacteria there. We keep them in check, and they're okay. But antibiotics and other foods can kind of wipe out your microflora in your gut, and you have to like kind of regrow it again. We have our mucus from goblet cells, and they help as well with bacteria. We also have secretion of IgA antibodies in here as well, which also help us prevent bacterial overgrowth as well. We don't want that to go crazy. Have you guys heard about they do fecal transplants now to help people, especially with C. diff and other things? What they're finding, though, is that some people, I don't know if it's true or not. This is an anecdotal story, but a patient was very thin, needed a fecal transplant. They got it from an obese person, and then they got obese. So they're blaming it on the fecal transplant. I don't know if that's true or not. Maybe they just ate a lot. Who knows? But things that are in the feces, these other things may cause problems in the next person. So, again, large intestine, fluid, electrolytes. Most of the fluid absorption occurs in the small intestine. That last little bit occurs in the large intestine. Not all the water is absorbed. Some of it is left with the feces. There is some effect of aldosterone here. Not a major one, but remember the major thing for aldosterone is to bring water back into the plasma to increase your blood volume, to increase your pressure. So aldosterone here can help bring more water in for absorption and reabsorption to increase your blood volume, increase your pressure, and bring it back up again. Defecation, as material passes to the rectum, the pressure in the rectum increases. The internal anal sphincter here, same as the urethral sphincter, under the same principles. Parasympathetic is going to cause constriction of it. Sympathetic is going to cause relaxation of it. They're here as well when the need to defecate arises. The external sphincter is our voluntary control. That's what you can do to relax, to go to the bathroom. So your internal is your involuntary method to stop voiding. The external is the voluntary method that you can try to stop voiding. And then obviously you go to the bathroom, everything relaxes, and the feces passes. During defecation, the longitudinal rectal muscles contract to increase pressure as the anal canal relaxes. And this is aided by contraction of abdominal and pelvic skeletal muscles, and that is what your Valsalva maneuver is doing. So we have you bear down. We're activating the Valsalva maneuver, and this is what we're doing. Okay. So composition of feces, what's in it? 75% of it is water. The rest of it, dead bacteria, fat, inorganic matter, protein, undigested roughage. So the joke about the corn kernels coming out later on because we can't digest it because it's cellulose. So it comes out later. And bile pigments. Okay, bile is in here as well. We're going to talk about bile when we come to the liver and how we recycle it. If you're ever curious about what does your poop tell you, there's a Bristol chart, and you can say, oh, if I'm pooping out like little nuggets, what does that mean? Hard to pass. Then obviously type 7, watery, entirely liquid. What's going on? Obviously, this is fast transit time. This is slow transit time. Okay, you're pulling out more and more water. Okay. Flatulence, gas is a normal byproduct of the food we eat. Our bacteria eat all this, and they're going to release carbon dioxide and methane. Methane is what makes the farts smell bad. But all produced by your bacteria in your gut. And then you expel it. Smell comes from mercaptans, the hydrogen sulfide. If you eliminate gas through the mouth, it's burping or belching, and through the anus, it's considered flatulence. Those are the medical terms for it. Okay, those are all the main tube of the system. Let's take a break, come back at 2:35, we'll do the accessory organs. Okay, we'll finish up. I misspoke before regarding ghrelin. Ghrelin is the hunger hormone. I said before it was satiety. So ghrelin, when it comes out, tells you you're hungry, you got to eat something. Leptin tells you that you're full. Okay, so just correct those in your notes. Ghrelin stimulates appetite, makes you hungry. Leptin tells you that you're full. Okay, so moving on to the liver, largest abdominal organ, right beneath that diaphragm, mostly on the right side. Some of it does creep over to the left. Very strong regenerative capabilities for the liver. Due to mitosis of the hepatocytes, they're continuously undergoing mitotic division to regrow. So much so that with a liver transplant, you can do a living donor. You can donate like half of your liver. Not all of it grows back, but some of it will grow back for the donor for transplants. The structure of the liver is very unique because of what it does with the portal system. It mixes arterial and venous blood together as it goes into the liver. They have these hepatic plates surrounded by these capillary sinusoids, very permeable, blood protein, fat, and cholesterol. This is where we can get rid of proteins and make proteins. It happens in the liver. So if you look at this picture here, you'll see here's the portal vein, here is the hepatic artery, and here is your bile duct. Okay. When the portal vein, it mixes and comes in this way. The arteries also mix as well. That's why when you see a lot of this stuff with the liver, some of the colors are going to be almost purple. Purple because red and blue together gives you that mixing, so you'll see it as purple. It won't be so much red, it won't be all blue, it turns into a purple because we're mixing arterial blood from the hepatic artery and the portal vein blood from all the Splanchnick system. They mix, we get a combination that they turn a little different colors. We also have these sinusoids, and that's what these things are here. The space is just talking about these cells right there. What these cells do is they are the ones who create bile. So I'll change to green so that bile comes out, goes into the bile duct, where eventually it will turn up and go into the gallbladder. So this bile will come up and then it just gets stored in the gallbladder and starts to concentrate in the gallbladder. So the bile that the liver produces isn't very strong. It's got to get stored in the gallbladder where there it starts to concentrate and get stronger and stronger as a chemical. And we also have our lymph system here in the white. Again, what's the purpose of the lymph system? Prevent edema by doing what with the proteins? Take them out of the interstitial space, put them in the lymph system, and then eventually it goes to the liver and we dump those proteins into the liver, we can get rid of them. But we get it out of the interstitial space to prevent edema formation. And the lymph will just drain into the biles and then we get rid of it that way. So our hepatic portal system, you can see here's our hepatic portal vein right there. And you can see all the stomach, large intestine, small intestine, they all branch and they all connect there into one hepatic portal vein, which then goes into the liver, mixes with the arterial stuff, and on the back end of the liver, we drain into the superior vena cava or inferior vena cava, and then we go back up to the body. Everyone good there? Okay. Here's our portal system again. Unique system of capillaries, veins. The total hepatic blood flow is about 25% of cardiac output. A lot of that blood goes to the liver. That's why if people ever had liver surgeries, they lose a tremendous amount of blood. They have to refill all that blood because of the blood they lost with the liver surgery itself. Lots and lots of blood through there. What is excreted by the liver into our bile? So we talked about how things are metabolized. If they're renally metabolized, we pee it out. If it's metabolized by the liver, we dump it in the bile. The bile goes into the small intestine, then it goes throughout the intestine, and we poop it out later. Endogenous or naturally occurring things, your bile salts. So urobilinogen, we'll talk about the bilirubin in a little bit. Lecithin. This is very big with gallstones, the lecithin. And bilirubin. No enterohepatic circulation. You'll see what happens with bilirubin in a little bit. Exogenous, drugs that are secreted in the bile. Your ampicillins, sulfonamides, penicillins, those are dumped into the bile, and it's how we get rid of these drugs. We excrete it later. So what does the liver do? Detoxification of blood. As long as your liver is working, your body will detoxify itself. There's nothing else you need to do for it. Does it automatically, you don't think about it. Carbohydrate metabolism, blood glucose to glycogen and fat, production of glucose from liver glycogen, or other molecules called gluconeogenesis. You're making new glucoses from amino acids. The liver does this for you. Lipid metabolism makes triglycerides and cholesterol. It also excretes cholesterol in the bile. And production of ketone bodies from fatty acids. So all this ketone production comes from the liver. Protein synthesis makes albumin, major contributor to what type of pressure in the body? Oncotic pressure. Okay, production of plasma transport proteins. Remember, a lot of these proteins, if they're fat soluble, they need a carrier to get around. They can't dissolve in the plasma, so we have these proteins. They bind to the proteins for a little ride. And our clotting factors. Okay, all those fun, factor 10, all those different factors, all come from the liver. So if someone had, and then finally secretion of bile, bile salts, and our bile pigment. These are the 5 functions of the liver. So if someone has liver failure, all of these things are going to be reduced. That's why the clotting proteins go away, the albumin goes away, they fill up with fluid. They may have issues with carbohydrate metabolism because they're not going to be able to store the sugars as they need, so on and so forth. So these are the different functions of the liver. Liver makes between 250 and 1,500 mLs of bile per day. Bile is made of bilirubin, bile salts, the phospholipid, the lecithin, cholesterol, and the inorganic ions. But again, it just stores it. So if you look at this picture here, the bile is produced from the liver. It comes down, all the way down. But when it comes down to here, what's closed? The sphincter of Oddi is closed. So this bile will back up again, and it works its way up, and it finally collects into the gallbladder. That's how we fill up the gallbladder with bile. And then once what hormone comes out, which relaxes the sphincter of Oddi? CCK, cholecystokinin. Sphincter of Oddi relaxes. The bile will pour into what part of the intestine? Duodenum, along with any pancreatic enzymes that come along the way as well, and then go in. Because here's your main pancreatic duct, where all the pancreatic juices, the bicarb, are going to come in, and they'll mix with the bile and help with digestion. Bilirubin, okay, produced in the spleen, liver, bone marrow, derived from the heme, from our hemoglobin. So when cells rupture, whatnot. Bilirubin, when it says it's unconjugated, that means it's free. Free bilirubin is floating around, not very water soluble, so it has to be attached to a protein. Because this is attached to a protein, can it get into the kidney? Nope. Or secreted in the bile. So it just kind of stays trapped in the blood. Bound to that protein, and it floats around. The conjugated bilirubin is bound to glucuronic acid. And when that binds together, it makes it water soluble. And because it's water soluble now, where can it go? Into the kidneys. And that's what gives the color, that amber color of your urine. It's just the bilirubin left over, among other things. Now, conjugated bilirubin, it's bound to the glucuronic acid, secreted into the bile, where it's taken to the small intestine. Bacteria will turn it into urobilin, which makes the feces brown. 30 to 50% is reabsorbed or absorbed by the intestine and taken back to the liver to make new bile. So this bile is a circuit. Some of it gets released into the feces, but some of it comes back into circulation, goes back to the liver to make more bile. Some is used to make bile, some remains in the blood, where it's filtered by the kidneys and gives it the amber color of urine, which is why when you are very well hydrated, you're peeing out a lot more water, dilutes all that bilirubin, and you get a clear urine. When you're very dehydrated, less water in the urine, but all that bilirubin is still there, and it gives it that amber color. So bile acids are derived from cholesterol. Most is recycled in this enterohepatic circulation. That's that cycle, how we can recycle bilirubin. 0.5 gram of cholesterol is broken down and lost in the feces through this process, this enterohepatic circulation. We can bring back bile, bile salt at least, from the small intestine and bring it back into the liver for recirculation. Blood glucose, okay, removes glucose and stores it as glycogen, glycogenesis, and triglycerides, lipogenesis. This is why when you eat too much carbohydrates, your body is going to store it as fat. Excess carbohydrates we store as fat. Breaking down glycogen, glycogenolysis, and puts it into the bloodstream, all that glucose. Makes glucose from amino acids, gluconeogenesis, and fatty acids into ketones. That's ketogenesis. So all these words. And this picture kind of just shows you with high blood glucose, insulin will cause increased glycogen storing, decreased gluconeogenesis. Insulin, we don't need new glucose being made. And decreases glycogenolysis. We don't want sugar going into the blood with insulin. The opposite is when you have low blood glucose, we get glucagon to come out. And we'll learn more about glucagon next week. But glucagon is the opposite to insulin. Whatever insulin does, glucagon does the opposite. Insulin lowers the blood sugar, glucagon is going to raise your blood sugar. That's why all the effects are the opposite when it comes to glucagon and glucose production. Also stimulates lipase in adipose tissue, so it converts stored triglycerides to free fatty acids and glycerol, which is lipolysis. Free fatty acids can go into the blood, used for energy by the skeletal muscle, but when you use free fatty acids, you're going to make ketones. This is where the Atkins diet comes into play, where you don't have any sugar at all. You don't have any sugar, you're not using glucose that is an energy source, you got to get it from somewhere else. So you start burning down fat. Well, when you burn down all this fat, you're going to produce all these ketones. So people who are on totally cut out carbohydrates, they may have like a fruity breath because of all the ketones, produce a fruity smell to their breath as you blow off the acetone or the ketones. So they have a different breath to them. That's one of the people who have like diabetic ketoacidosis. Too much ketones, their breath is fruity because of the ketones they're exhaling. In the liver, glucagon stimulates the conversion of fatty acids into the ketone bodies to be used as an energy source. So when you get excessive catabolism, what does catabolism mean? Break down. What is to build up? Anatomy. So ana is up, tomi means to cut. So anatomy is to cut up the body. That's how anatomy word comes from. And so anabolism is building up, catabolism is breaking down. Transport of lipids in the blood. You've all heard of HDL, LDL, IDL, VLDL, whatever. The big thing is this. All these atherogenic proteins are going to cause your body to build clots and plaques in the arteries, all this extra stuff. These are produced from the liver. And if you notice, they all have this little hat on them called ApoB. ApoB is an apolipoprotein and kind of is a marker for the liver to know that these need to be taken out. We also have reverse cholesterol transport. This is your HDL. This is the good cholesterol. We want that to be as high as possible because HDL is like scavengers. They take all this extra cholesterol from the body and they bring it back to the liver to be taken out so we can lower our cholesterol, or the bad cholesterol at least. HDL you want as high as possible. Unfortunately, there's only one way to get HDLs up. Exercise. That's it. They tried a drug a few years ago to raise HDLs with a drug. The problem is it caused a lot of liver problems. They had to pull it from the market. So HDLs, you want to get it up higher, you got to exercise. But HDL has the apolipoprotein A on it. It's like a little hat. Again, tells the liver, these are our scavenger proteins. We want to bring these in. HDL will clear our body of cholesterol. And finally, we have our albumins with transport, oncotic pressure, globulins, transport, blood clotting, and then our clotting factors as well. These are all done by the liver. So again, if you have liver failure, all these things are reduced because you aren't making the proteins, you're not making the cholesterol, and all stuff like that. Detoxification of blood. We can remove hormones, drug, other substances in 3 ways. 1, just get rid of it in the bile. As we're making our bile, we can dump stuff in there, different antibiotics go in there, and so forth. It can be phagocytized by our Kupffer cells. The Kupffer cell is the immune cell in the liver. Okay, what's the immune cell we learned about in the brain? Microglia. In the liver, Kupffer cells. Or we can alter them by our hepatocytes. So ammonia, which is not a very good thing to have in our body, is converted into urea. Porphyrins are into bilirubin, and purines are into uric acid. This urea goes back into the blood, and our kidneys can get rid of urea, no problem. Steroids, xenobiotics are altered and then secreted into our bile for metabolization so we can get rid of them. Now, ammonia. If someone has liver failure, are they going to be able to convert the ammonia to the urea? No. So they're going to have a buildup of ammonia. That will cause altered mental status, a lot of other problems with these patients. They need to be brought to the hospital and given a chelator. Talked about that before with potassium. Chelator to get rid of the ammonia. It's usually called lactulose. If you've ever heard of lactulose, that's what they use to get rid of all the excess ammonia in those patients. That's the liver. As long as you have a healthy liver, it's going to detox everything for you. Just give it a chance. Gallbladder. Sac-like organ attached to the inferior part of the liver, stores and concentrates bile from the liver. That's all it does. Just stores and concentrates it. Function, emulsify the fats in our food, helps with absorption of fat. We have bile salts and IgA antibodies that inhibit growth. Neutralizes the gastric acid in the intestine as well, and aids in the excretion of bilirubin. We just put it in the salt and we can get rid of the bilirubin that way. So in this picture, we have our fat globule at the top, and we have our bile salts, and they start to attach themselves all the way around it, and that is enough to emulsify that, the fat and get it into an absorbable size. Hormonal control of biliary secretion. Obviously, we get food in the stomach. That's going to cause distension, and that starts the process. Vagal stimulation causes weak contraction of the gallbladder. But what do we really need to be released? cholecystokinin. And that gets released into the bloodstream, which then goes to the liver, goes to the gallbladder, and tells it to start to contract, and tells the sphincter of Oddi to start to relax. So that bile can leave, pancreatic juices can leave, and all go into the duodenum. Bile is about 15 times concentrated than it is when it's produced. That's in the gallbladder. Secretin via the bloodstream will stimulate liver ductal secretion to make more bile as well. So that's where that comes into play. So cholecystokinin is the big one. Secretin is there a little bit as well. Any questions about gallbladder? Very simple structure. It doesn't do much until it gets aggravated. You got to get it out. But otherwise, it does its job. Pancreas has endocrine and exocrine functions. The endocrine functions we're going to talk about next week when it comes to like the islets of Langerhans. But the exocrine function, which are these acinar and ductal cells. So if you look at the islet, the yellow cells on the outside are the exocrine glands. The other ones are going to produce all the pancreatic juices. The ones on the inside are the ones that make the insulin, which we'll talk about more next week. Okay. So, pancreatic function. Obviously, the big thing is bicarb to neutralize the acid. We need that. The other thing that the pancreatic enzymes have in it are these digestive enzymes. Proteins, trypsin, chymotrypsin. They're going to break down these proteins into smaller and smaller units. Carbohydrates, pancreatic amylase. To break down lipids, we have pancreatic lipase, cholesterol esterase. So pancreatic lipase is going to break down for fats. Amylase, just think of carbohydrates. Amylase is for carbohydrates. Lipase is for the fat. And we also have secretion of trypsin inhibitor, prevents autodigestion of the pancreas. We don't want the pancreas to digest itself with all this alkaline fluid either, right? Because the pancreas is producing all this bicarb and all the other digestive enzymes. They want to make sure that the pancreas itself is protected from all that bicarb as well, which is alkaline. So we have this trypsin inhibitor, which protects from autodigestion. This, you don't have to know this one, just kind of explains again how we get, here's our lumen, here's our chloride, there's our water and the bicarb. And the water, here's our bicarb getting dumped into the lumen to help with digestion. So regulating pancreatic secretion, going back to these hormones, acetylcholine, parasympathetic nervous system. It makes sense that will be released because we're rest and digest. Cholecystokinin, makes sense, rest and digest, stimulates enzyme release. Secretin, large amounts of water and bicarb, rest and digest, it's going to come out. Secretin has 3 different phases. Don't worry about that so much, just understand what secretin does. These cephalic, gastric, and intestinal kind of coincide with the different phases of the stomach, where first you smell the food, you start getting excited, things start moving, so you start getting your body ready for the food that you're about to eat. So, autonomic function of the parasympathetic division, while eating, and stimulates the release of insulin. So the parasympathetic division will get insulin out, ready to go for the glucose that you're about to eat, or the sugar. The sympathetic division inhibits insulin secretion and stimulates glucagon secretion. Sympathetic division, fight or flight or rest and digest? Fight or flight. Where do you need the glucose? In the blood and the muscles. Now I'll read that again. Inhibits insulin secretion, so the glucose is going to stay in the blood, so it can be brought into the muscles for action. And stimulates glucagon secretion. What's glucagon going to do to your blood glucose levels? Increase it. We're dumping glucose into the blood where we need it for fight or flight. And that's where the autonomic neurons for the pancreas come into play. And we're